how is mRNA delivered to cells? Previously we've looked at what RNA is, how we know mRNA codes for the desired proteins we want. So now we're looking at how we actually get it into our cells, actually get it to those target cells and make sure it presents the COVID spike protein or whatever protein or functionality this RNA has within our cells. So mRNA needs to be protected. If we directly inject it into our bodies, it will degrade immediately because there's enzymes in our extracellular matrix. Also, there's other environmental factors that may degrade this RNA. So really we need to encapsulate it in something. By encapsulating, we can target. This is really important when we consider the design of our mRNA nanocarrier. Again, falls back to the previous series on the size, shape, charge and coating of our nanoparticles. By optimizing those, we can optimize this delivery of this mRNA to our target cells. In particular, we should look at the nanoparticle charge and the nanoparticle coating, as they're the ones that will really impact where our particles will go, especially when we consider we're using a liposome nanoparticle, which we'll talk about a little bit more later. So briefly, RNA codes for proteins. Not all RNA codes for proteins, but the ones which we're interested in do. And in this case, we're looking at RNA that codes for this COVID spike protein. So this is produced by using a DNA template, RNA polymerase and nucleotides. RNA polymerase performs this from DNA to RNA by pulling in these nucleotides and forming this long messenger RNA strand. And then when this mRNA is delivered to our cells, it performs the conversion of this mRNA sequence to a protein structure. So why do we need to protect the mRNA? And that's something I touched on a little bit before, where mRNA will be degraded in the extracellular matrix due to enzymes. So it's fundamental that we have a nanocarrier. There are currently 9 FDA-approved RNA treatments on the market. And by using a nanocarrier, we can increase the bioavailability of the mRNA. As mentioned, without it, it will degrade. We also need to consider, though, where our mRNA is going to enter the body, as both from how the mRNA functions, also decreasing the invasiveness of the injection, so therefore more people are more likely to have the jab. And by using a nanocarrier, we limit the removal from the microenvironment where we've injected our liposomes, as well as we can optimize the liposome or the nanocarrier to uptake into our target cells. So how is our mRNA protected? So Liposomes are essentially our cell membranes, but stripped of all of these surface protein coatings and such. And they make brilliant nanocarriers, as they are like our cells already. So it doesn't really elicit an immune response. They're very easy to create, as all we need is lipids and we can disperse them in a solution of our mRNA. And then the liposomes can self-assemble with mRNA on the inside. Currently over 30 FDA-approved liposomal treatments on the market. And liposomes have been used for delivering RNA, proteins and other drugs which may be insoluble or generally difficult to get the bioavailability in the target location. So typically mRNA liposomes are produced using microfluidics. This is very similar to the droplet formation which I simulated previously, and this involves a discrete phase which contains our mRNA and a continuous phase such as water where lipids aren't immiscible, so they don't mix, they cannot freely disperse within the fluid. They form a small particle which is our liposomes, and that's simply due to the charges on the lipid. So the lipid has a positively charged head and a non-polar tail. The non-polar tail is very hydrophobic, whilst the head of the lipid is very hydrophilic. So they self-assemble, forming these small liposome balls or micelles. And using microfluidics, we can actually control how these flows come in, and we can do it in such a way that we can control how much mRNA is getting encapsulated, as well as the size of the liposomes, and ensure that they are forming liposomes as opposed to micelles. The exact method though is very difficult to know, given that it's IP protected. So RNA naturally is attracted to lipids due to the negative charge of RNA and the positively charged head of the lipid, as we mentioned earlier. And due to the way the lipid self-assembles, we can actually control the different lipids that are in this suspension. So we can add ones which may have a lipid PEG linker, so PEG being polyethyl glycol, which is a very good polymer for evading the immune system, as it acts as an invisible barrier as such. So to conclude, nanotechnology really aids us in delivering mRNA to the right cells and the right environment so that we can express proteins such as the COVID spike protein. Without nanotechnology, such as nanocarriers, in particular liposomes, we wouldn't be able to deliver mRNA. Liposomes are a tried and tested nanocarrier, and RNA is a tried and tested treatment. However, this is the first time that RNA has been used as a vaccine, but that's really not a problem, as we've clearly shown before that mRNA can code for a particular protein, and this is simply just changing that mRNA sequence to form our COVID spike protein in this case. And it's really exciting to see the developments of this, as with more nanotechnology such as mRNA being produced, we probably will see more of these mRNA vaccines appearing. And this is simply due to that mRNA is much easier to produce than proteins. Although cell-free methods for producing proteins exist, they're still difficult to mass produce compared to mRNA, which just requires that template DNA strand, RNA polymerase, and those nucleotides to mass produce messenger RNA. Thanks for watching, and see you next time.